All right, well, welcome to the capstone session of the May Institute. You've all, I guess, not just checked, and you've all been through at least some of the prior sessions. Some of you, I think, have been through all of them. And this session is going to attempt to challenge you. It may challenge us as well, but to bring that all together, to bring the skyline parts and the statistical parts together and analyze some real-life data sets that were created, some in the McCoss lab and a lot of them in Zurich, where our first speaker, Rudy Aebersold, is coming from. Your instructors are, here now, except for Mike, Ben Collins from Rudy's lab at ETH. And he'll be up here in a second. And then Rudy Aebersold from ETH Zurich, very well-known in the field, pioneer in a lot of what we've studied. Certainly inspiring to me in starting Skyline, and has been a great collaborator in targeted proteomics and DIA. And we're gonna focus entirely on DIA and swath. Or that's the same thing, two synonyms for the same thing. We're going to be focusing entirely on DIA swath in this session. Some stuff we've tried at other sessions, and some we're going to try here for the first time, and it'll be interesting to see how well it goes. If for those of you who have slower computers, slower laptops, you may have to buddy up, and we'll check your laptop capabilities soon. But now I've digressed from. We got Rudy Aebersold, and he's here, and he's going to be the next speaker, and then also Mina Choi in the back, who's our MS Stats expert, is going to help us take this all the way through to statistical inference on these data sets that we're going to be working with. We're still busy making flash drives. Can I get a sense of the kinds that the power of the computers people have brought? How many people have how many people have a laptop with 16 gigabytes of RAM? Wow, not so many. 8 gigabytes of RAM? Do people know? Okay, we'll go over this later. Forget it. So we're not actually going to get into, despite the schedule saying that I'm going to be doing hands-on, we're not going to get into hands-on until tomorrow. So anyway, we'll come back. We're working on flash drives. We'll have you all copy them. Do people have any idea of how much free space they have on their hard drive? That's pretty easy, right? Do people have at least 50 gigabytes of free space on their hard drive? Anybody not have 50 gigabytes of free? Okay, if you don't, start making some space. And we're not even going to make you copy very many raw files. So, the thing about DIA and swath is that it requires the data files are quite large, so we have some toy data files that we're going to let you play with, and then some fully processed files which actually get to be fairly large as well. So you're going to need disk space. If you don't have very much memory, toward the end you're going to have to buddy up, but for the first day you'll probably be okay. But anyway, so there's going to be hands-on, there's going to be talking, and we're going to focus entirely on DIA swath, ranging up to these large-scale experiments looking at the whole proteome. And to give us an introduction of what can be done, what has been done, and where we're looking in the future for DIA and swath. I'm going to let Rudy Aebersold take it over now. So.
Okay, thanks, Brandon. I thought you would end up with like a sales pitch and say, if you don't have 16 gigabytes and or storage and hard drive, yeah, I have something to sell.
That's a good idea. That's why you're better funded.
Okay, thanks for giving me the opportunity to talk here. I would like to start out by acknowledging Olga and her lab, and her group, and also Brandon and all the other instructors who put a lot of effort into these courses. It's actually a huge amount of work to get this organized and to get a coherent plan, and to do this for 2 weeks is a real accomplishment. So I think it's great. So this whole course is about, well, this capstone is about data analysis of, as Brandon said, DIA or SWOT data, and I will talk for a while about some considerations of this data. It's various points. I'm coming to them. What I'm going to cover, and I try to put the experiments we're doing in this technique a bit in the context of what we would like to learn, because oftentimes, surprisingly often, it is not really well defined what someone wants to do with an experiment. They say, okay, let's sequence proteins or let's quantify proteins without having a clear idea where this leads. So I spend a little bit of time on kind of the biological environment and the surroundings of it, the objectives. So you might also ask, why is this course entirely focused on data analysis? And when we first applied for funding for such courses, not for this one, but for courses, similar courses in Europe, we got rejected by the reviewers, and they said we need to offer a course that covers also wet lab and wet lab expertise, and we need to do something on the machine. And we always argued that this is not so. That you can separate this, because and this is probably the most important part of this whole workflow, is the data analysis, because many studies fail at this level. And this is a relatively old analysis where which highlights this point. So this was a very simple experiment. This was in data-dependent analysis, so in shotgun world, where someone sent around a sample of 20 highly purified proteins, and they were equimolar, pure, and only 20 in a tube, so far from being a proteome. And what they asked people to do is to use the techniques they had in their laboratory. So this is not so long ago, it's like 10 years ago, not even that, and to report back what they had found. And the results really were quite abysmal. I mean, there's many that found none. One found about 1,000, and so it was clearly something wrong. And then I was asked to review this paper, and then I suggested to separate the parts apart in the various parts. I mean, we did not need to talk about the sample. The sample was defined and had a ground truth, as far as one can say. But then I suggested one would separate the mass spectrometry part and the data analysis part. So the consequence was that all the data were sent to one place, and someone who really knows what they're doing with this data analysis, and then they analyze the data under consistent conditions. And it turned out that even that from the data collection, most people were actually able to generate data which were covering most of these proteins, but they couldn't figure it out using the software tool. So that's why we then eventually drew up this graph and said, well, to get to a result, you need to cover many mountains, and some are easy to do, and some are very difficult to overcome for many people. And this is the informatics part, and that the data analysis, and that clearly, I think, is true also for in general, and certainly also for DIA analysis. So that's why it's a rationale. We'll focus not so much on how you digest the protein, although that's of course also important, but on the data analysis. So that's just justification of the topic. So now I'm gonna. I thought I would talk about 4 points. They're listed here, and I go now through one by one. The first one I would like to discuss is how does proteomics fit into experimental biology, and I'll make a few comments about that. So you all probably know that picture. It's a historic picture from the year 2000, when the human genome sequence draft was announced, and with that came a lot of predictions from the scientists who fed these lines to the politicians, of course. It said that this the draft of the human genome sequence would allow scientists to alert patients that they are at risk for certain disease. I think I should use the cursor here. That they could reliably predict the course of disease, precisely diagnose disease, develop treatments of new molecular level. So basically they said, in 2000, if we have a sequence of a person, we should be able to look up what this sequence tells us and to make projections into the future. And this, of course, has not happened. And basically they said the sequence would explain how biological systems function, whether someone develops a disease, how this disease can be cured, and so on. And this, of course, did not happen. But what did happen is that the new techniques were developed to allow scientists to very quickly and relatively cheaply sequence whole genomes and transcripts. And so this is a very famous graph from the NHGRI, which says that the cost has been dropping faster in the large-scale genomic sequencing than Moore's law, and it actually continues to drop. And the projections are that soon we'll be able to identify or sequence a genome for about $100. That's projections of the specialists. So it's probably less than many people spend in a month for coffee. So the real revolution of the genomics is not so much the initial sequence draft, although of course this was also very informative how this genome is structured, but it came from technological revolution to rapidly and accurately and cheaply sequence, basically measure thousands of genomes. This led to very large genome-wide association studies, transcriptome maps, and so on, and is very widely used in the hospitals. So what about proteomics? We tend to always kind of link behind a few years in genomics for technological achievements, but in 2000 and I believe it was 13, 2 papers were published which said they would present a draft of the human proteome. So this is the analogous claim that was made for the genome a bit more than a decade earlier. And these both papers were published back to back in Nature. So there's two things I want to comment on about this stage. So this was clearly the title of these papers clearly said that we had reached in proteomics a stage that was reached in genomics a decade ago, where we would be able to, we know basically how a proteome looks like. And then the question was, is this map, even if it's accurate, actually useful for biologists, because the sequence of the genome was marginally useful for understanding biology. It was useful for many things, but not for understanding biological mechanisms. And so the question would be, would this draft, if it is actually a draft and roughly correct, would that be useful to support biology? Okay, so I come to these two questions now in the following. Here is simply a indication how rapidly this field has evolved. In 2000 or even 2005, there's almost no data generated with current standards. And all these data were generated incidentally by data-dependent analysis, shotgun proteomics. And then we actually started a project which we call the Human Peptide Atlas, where we asked people who are out in the field to collect data, well, then send the data over when they publish a paper, and we would do an aggregate analysis. Now there's several of these repositories, and one probably the most widely used is Pride, and also Mike McCoss has one, a very large one, Panorama. So these are extremely useful resources because there's millions of dollars worth of data that have been collected in some context are available for scientists to re-explore. So now this database, which is a collection of human sequences, there's millions, hundreds of millions of spectra, are in there, we ask them if this is analyzed in an aggregate way, what part of the proteome does this cover? Remember, in 13, these two papers came out and said we basically have covered the human proteome. And whatever anyone did, it saturated out at around 14,000 proteins. So this is a peculiar situation that some people published a paper where they said we have basically covered the whole proteome as a map, and the public effort which combines basically the majority of the publicly available data cannot reach that level. So the level is about 14,000 proteins, 14 to 14,500 proteins, and this is short of about the 20,000 open reading frames that are thought to be in the human genome. So the question was, where are discrepancies? Why are these numbers different? And then a lot of effort was spent to reanalyze all this data together, because it is an important question whether we can say we basically know how a proteome looks like to a mass spectrometer, or whether there is roughly a quarter of the proteome yet to be discovered. I mean, it has technical implications, but also biological implications. Of course, whether we know how to measure all proteins or whether we're not able to. So a number of groups who have been very active in this field of developing tools, analysis tools and statistical tools, they're listed down here, including Jürgen Cox and Matthias Mann, Ron Beavis, Alexei Neshvisky, Gil Omen, took all this data that were in these draft papers and analyzed them either together, either by themselves or together with the aggregate data that people had donated to the public domain, this Peptide Atlas project. And what they found is that either the data sets by themselves or in combination with the public information capped out around these 14,000 plus proteins, exactly the same or very close to what the public effort was doing. So what we think and what's happening is that these papers over-predicted the number of proteins identified from the evidence, the spectra available, and I will come back towards the end to describe why this happened and which part of the data analysis they probably did not sufficiently control. It's an explosion or an aggregation of false positive identifications in very, very large data sets, and we also need to be very cognizant of this issue in the DIA world, and this has actually been addressed to a large extent, and I'm sure this will be discussed. So now, just a side note on editorial policies and how journals function. So we attempted to publish this summary where the data from these individual papers had claimed essentially complete coverage of the human proteome and their relationship to the public effort. We tried to publish this as a commentary in Nature, because we thought it would be useful to know for people who are beyond the proteomics field that in fact these papers likely overestimated the number of proteins published, and to describe the reason why. And then this was eventually rejected as no longer interesting because the editor said this is the papers were published a while ago. I mean, it took about 2 years to actually do this reanalysis, and they eventually said it was no longer interesting, but it is interesting because there is now 5,000 or so proteins in the literature which are claimed to have been identified by mass spectrometry, and we think they're not. So someone will now use this data and make claims of things propagate in the literature, and but Nature was not willing to consider this seriously. Okay, so that much to the effect of coverage. So if we talk about proteomics, we still are unable to claim that we can identify every protein in a proteome, and it caps out around this. The coverage of the human species caps out around these 14 to 14,500 proteins, and what the rest of the proteins are doing, whether they are expressed, why we don't see them, is actually an interesting question, and there is whole groups trying to figure out why this is. Is there some technical bias? Is there biological bias? Or some kind of combination? But from the purpose of this discussion here, this course, I will now go back to the analogy of genomics, where really the biological and clinical revolution happened not from the looking at the initial sequence, the draft, however complete and correct this was, but from the ability to remeasure many genomes with relative ease. And I think this is now also happening in the field of proteomics through the techniques we are considering in this course. So I would think that we can claim that there's now this proteomics revolution is happening because the techniques are quite mature and are certainly of a reasonable throughput, actually a quite high throughput, but they're not able to cover the whole proteome. This is something that we should keep in mind. But it is possible now to cover hundreds, if not 1,000 proteomes through these DIA techniques in very high throughput and relatively good reproducibility and quantitative accuracy. So now let's assume we can do these measurements. We can either use the data-dependent, the shotgun methods, and map out most of the proteins in a sample, or we can use these DIA methods and do basically large-scale data sets of cohorts, like let's say clinical cohorts. Now I would like to discuss briefly how these results how can we imagine that these results are being used to learn something biologically relevant. So if you work, I don't know, so who is a clinical or biologically oriented scientist in here? Okay, so probably half to 2/3. So I would assume that most of those work in some form of a research group or department, or university, and that this university might have a department of genetics, cell biology, if it's a basic science department, and maybe structural biology. So all of these units, departments, or research groups, they have in their name and the way they have traditionally operated, they try to assign a function to a specific protein or protein complex. I think most significantly is this thinking is presented in the structural biology units, where you try to isolate some molecular machine, let's say a ribosome or a transcription factor, and you try to say that this factor or this protein or this complex is doing this particular function. Ribosome is making proteins. A transcription factor activates genes. So for that, it is important, of course, that if you pursue this research, it is very important that you can identify this protein, you can quantify it, you can determine its sequence, and eventually also its structure. And for that, I think these DDA approaches, the data-dependent analysis or shotgun or discovery proteomics, has been tremendously successful because they can identify with relative ease now most of the proteins in a sample. So if you want to identify some secreted protein for which you have a biochemical assay, for instance, whether it is able to stimulate a cell for growth, you can sequence the proteins in that sample, and you're very likely to find it, and then you simply have to see which ones among these proteins has this function. And this is usually then a biological assay that is being set up. So the approach, the question in this, which has actually dominated proteomics till extremely recently and to some extent today, the question would be, can we discover a protein that carries out a specific function? And for that, the best technique is discovery proteomics, clearly because we can discover such proteins. And the approach was to discover as many proteins as possible and find in that large number of proteins, 10,000, 12,000 proteins, the one that really matters. It matters in the sense that it carries out the function we would like to study. So now let's go over to what we are really discussing here in this course, but also in many different contexts. This is the system biology or data-driven research, which has becoming very prominent, and it is used also in many research institutions, of course, There's some research institutions which are almost entirely built on this principle, like the Sanger Center in the UK, like to some extent the Broad Institute here, who have fantastic platforms to generate massive amounts of data of very high quality. So this would be genomics, transcriptomics, increasingly proteomics, metabolomics, and so on. And what is the expectation here? We don't try to assign a function to a specific protein or gene or mutation. We basically ask a different question. We would ask, how does the system as a whole, the cell or the tissue or an organism as a whole, react to some perturbation? This perturbation might be in, for instance, if you think of cancer genomics, might be specific mutations that occur in cancer cells but not in normal cells. And so the question is not what is this mutation doing, but the question is how does the cell, the body, react to the presence of this mutation, and how does the body react, or the cell react, to become a cancerous cell. And the expectation is that we would discover the role of a protein or a gene or a mutation in the context of the overall molecular makeup of the cell. And the techniques here would be that we cannot just do this once. We cannot just take one cancer patient and do a molecular analysis. It's usually largely useless, or not informative, but then you can build cohorts of hundreds of patients which have similar phenotypes or identical phenotypes or disease. Then the expectation is we would be able, from these patterns, genomic, proteomic, metabolomic patterns, we would be able to learn something about the underlying biological mechanisms. So the question we're trying to answer here, and I think this is the way I would say these DIA techniques are best positioned, is to do serial measurements on related but different samples, and then ask how does the cell or some other biological system react to an insult, for instance a mutation, a gene deletion, or also an environmental impact. So it's really two different questions, and for the first one, discovering factors, DDA is great for finding out how the systems react. DIA is great, and as I will show, and then later on I'll try to indicate that neither by themselves is probably sufficient, and we need kind of a combination of both. Okay, so if we adhere to the view that in this that on this side here, this data-driven research proteomics has a role to play, then we would need to ask what data do we need to generate, and what is the properties of this data that are really useful to support such data-driven analysis. And so I think certainly the picture that we pursue in our group is that of a data matrix. So we're no longer interested in having a list, an inventory of proteins that are present in a cell or in a tissue in a species. This is also useful, but we are not interested in a list, but we're interested in a data matrix where we have a number of samples. This could be patients that from a clinical cohort. This could be a time series of a drug response study. It could be cells where specific genes have been mutated or knocked down or knocked out. And we would like to measure how does the system react. For that we need a number of samples. And we would like to observe a number of proteins, ideally all of them, but we can't. But we would like to do this very reproducibly, that the matrix basically has very few, if any, missing values. And the values that we here, so for instance, that protein measured in that sample should be an accurate quantitative value, and we'll come back to that also on Friday, why accurate quantification and reproducibility is so important for this data-driven analysis. So the picture I think you might carry forward in this capstone module is that of a data matrix, and you should ask now how we build such data matrices and how do we use them then to learn something biologically important. It turns out that the only methods that can deliver in proteomics that can deliver such matrices which are quantitatively accurate, have few missing values, and basically are generally reproducible are the techniques we talk about here. The DDA methods cannot really do that. Even with great effort over more than 10 years to standardize samples, sample processing and data analysis and mass spectrometric method, this has not really been achieved. With DIA techniques, this is very easy to achieve and is achieved in many laboratories, as you will also then see why it is fundamentally a different approach and why it has properties that are well suited to support such data matrix generation with these DIA techniques. So this is now just to indicate in the reality with real data how these two different techniques, the discovery technique and this DIA technique, perform in this context of generating a data set which we represent as this data matrix. This is not data from us, this is data from a spin-out company, Biognosis, who published this, and I will explain now what this slide shows. I think it's a very graphical slide and very impressive, and it will illustrate why for the generation of this data matrix, why we want to use DIA methods and what their performance roughly is. So what they did here is they took exactly the same sample, a lysate of a complex human cell, and they shot this lysate a number of times, like 24 times. There's 24 repeat technical analysis of the same sample in an instrument, a liquid chromatography instrument with a mass spectrometer. In fact, in this case, it was a trapping instrument, a Q-exactive, but it doesn't really matter. And they performed this analysis on the same instrument with the same sample in the two modes of mass spectrometry that I briefly discussed, the discovery approach and the DIA approach that is suitable for generating these data matrices. So they call the DIA hyperreactive monitoring, HRM. Don't worry about the terminology. The terminology issue is a stupid one, but it's all the same. HRM, swath, DIA is basically the same thing under different disguises. Okay, so what do we see? Let's first look at the graph up here. We see on the y-axis the number of peptides that were identified in sample 1 or technical injection 1, and then the samples, the peptides that were identified in technical injection 1, 2, and 3 cumulatively, and then all the way to 24. So we see that the number of peptides identified stays nicely up there, so there's no attrition or very little attrition. So what that means is that the same peptides were basically detected in all technical repeats. And so we knew when you want to build a matrix out of this data, that's what you see. You see the number of samples, you see the number of proteins here, and you see very few gaps here. This is the same, basically a graphical representation of the fact that these peaks here stay up, so that the same protein, same number, and same type of proteins are identified, or peptides are identified in repeat injections. This is different when you look at the blue curve, where the same samples on the same machine was analyzed by DDA, shotgun mass spectrometry. You see that it identifies also a large number of peptides, although not as high as the DIA analysis. But when you ask how many peptides are consistently seen throughout for 24 injections, the number gets rather low, down to about 5,000 or so. So, and this is due to the fact that this technique, the DDA technique, does not has a huge number of peptides to choose from and does not choose always the same peptide. So you get this, you get a somewhat erratic measurement, not a wrong measurement, but a leaky measurement in the sense of such a matrix, where you see here there's a lot of missing values, and the number of proteins that are really, or peptides are really detected throughout is relatively low. And when you now, of course, when you think that you want to do some kind of machine learning or clustering or so, you can appreciate that if you had such a data matrix, the results would not be as good as if you had a data matrix which is rather complete. And the other point which is also apparent here is that the HRM, the DIA technique, has a lower coefficient of variation, so it's quantitatively more accurate than the shotgun method. which again is beneficial for downstream analysis of these matrices. Okay, so what type of research is used to work with such quantitative protein data matrices? This would be biomarker studies, genome-wide protein association studies, where you try to link a protein variant or protein abundance to genomic locus. I'm coming to that probably on Friday, or even today a little bit. And then if you want to identify pathways or clusters or networks, these are all, or population-based studies, these are all types of studies which will benefit from having a data matrix that has a very large number of samples, quite quantitatively accurately measured. So this was what I wanted to say for the first part. And what I hope you appreciate, and it's a very short summary of this part, is that there is, from a biological point of view, there's different objectives people want to achieve. Some have to do with discovering a specific protein that carries out a particular function. Others have to do with learning from the behavior of a cell. And for both of these approaches, broadly speaking, there's different methods that are optimally suited for this purpose. So it's not that one technique is better than the other. It's simply they serve different purposes. But for the DIA method, that we will not discover new proteins a priori, but we can see how these proteins behave quantitatively under various conditions. Okay, now if we accept that our kind of guiding light is such a data matrix, I would now like to discuss how, if we have a limited budget in the lab, which is the case probably for most of us, how should we invest this? How should we invest this money to generate data set that is optimally, where we have maximized our chance to learn something and eventually write a paper? Should we do many more proteins, really dig down into the proteome as deep as we can to get as many proteins identified as possible? Or should we try to do as many samples? Of course, you might say, well, do both. That's true, but that's where the limited budget comes in. We can't do both with a limited budget, because when we want to dig deep down into the proteome, it comes at a cost of money and time and resources. So we have kind of a trade-off between going very deep and going very broad in our data matrix. So this is the point I would now like to discuss a little, and this basically is the work from Ting, who's back there, and who is a PhD student in Olga Witek's lab, and a while back we discussed this question, and Ting has done a fantastic job of actually addressing this, and I think a very convincing way. So I hope I'm doing justice to your work and I talk about it now. So this is the techniques we have available in our proteomic portfolio. This is shotgun and discovery proteomics, where we detect by survey scans a very large number of peptides, and then the mass spectrometer will sequence one peptide after the other in a sequential manner. We have the targeting proteomic schemes like selected reaction monitoring or precision reaction monitoring, PRM, where we follow a specific peptide that is predetermined by the researcher based on its precursor mass and its sequence. So it's a needle in the haystack searching technique. And we search for that peptide to quantify it in a sample, and we can do this for a number of peptides concurrently. The peptide will definitely be detected and quantified if it is in a sample, because the mass spectrometer is instructed to always look for this peptide throughout the whole duration of the mass spec experiment. Incidentally, this is the retention time axis of the peptide separation. This is precursor mass axis that the mass spectrometer detects, precursor axis. And then we have now the DIA technique that we talk about here, that we would cover basically the whole range of this peptide field, area of retention time and precursor mass, and all the peptides are fragmented, they're fragmented no longer as individual peptides, but they're fragmented as packets. And so we generate composite peptide spectra from each of these packets, and then we have to deconvolute them after to learn how this precisely functions and what the technical pitfalls are. From a point of view of performance, we see that this technique the shotgun technique is very well suited and unmatched in going extremely deep into the proteome, as I showed, up to 14,000 or so proteins of a human proteome, and but it comes at a cost of tens or even hundreds of runs. So this 14,000 coverage was achieved with thousands of LC-MS runs, of course, which is expensive. So then the other technique, the SRM technique, can do many samples quite reproducibly, but only with a rather low number of proteins, let's say maybe in the range of 100. And this DIA technique now can do also many conditions, hundreds or now going up to into the thousands. And it can do it it consistently measures thousands of proteins. The typical standard today on a high-performance instrument, on a relatively, let's say on a cell type, on a homogeneous cell type, is in the range of 5,000 to 7,000 proteins. That's the state of the art. Okay, now what does that mean? We know, of course, that the cell, a human cell, a differentiated human cell, will express roughly 10,000 proteins. I mean, not 10,000 protein molecules, but 10,000 protein products of about 10,000 open reading frames. It expresses, incidentally, about the same number of protein molecules that there's people on Earth, which is an amazing number. About 8 billion or so proteins is present in a tiny little eukaryotic cell. So now, what does it mean when we say we can detect by DIA or any other mass spectrometric method, let's say 1,000 or 5,000 or 3,000 proteins? What I should say first is that when we identify proteins by mass spectrometry, there's always a bias towards the more highly expressed proteins. They're simply easier to catch by any method. So we generated this plot here. This is based on work from Martin Beck, who was a postdoc in our group, and he worried about things like what's the dynamic range of proteins, what's the concentration of proteins in a cell, and so based on results we generated this plot. So with you on the y-axis is a total protein mass that we think is in a cell, not the number of proteins, but the total protein mass, and we on the x-axis is the number of proteins that would be needed to identify or cover these respective total protein mass values. So what I'm saying here is we cover 5,000 protein, 1,000 proteins. The top 1,000 proteins of a cell will constitute about 94% of the total protein mass. So this is, of course, not a precise value. It depends on cell type to cell type. But this was HEC293 cells, which is a cancer cell line. So if we cover about 25 or 20 or 3,000 protein, we approach 99%, the vast majority of the total protein mass. That's based on the data that Martin Beck generated. Now, what does this mean? It means, of course, that there is many proteins in a cell that are functionally important, but they are not expressed at high levels. And these proteins we will have a hard time detecting, but from the total protein mass that is in the cell, they don't really matter. They matter, of course, biologically, but they don't contribute a lot to the total proteome mass. Now, the argument that we would make would be that if we are interested in discovering a factor that carries out a specific function, let's say a transcription factor, and we want to see that protein, we clearly are not sufficiently covering the proteome if we cover 3,000, 4,000, 5,000 proteins, because the chance that we do not detect this protein is very high. However, so then if we really want to measure this protein, we would want to do very deep analysis by DDA. However, if our question is how is the cell reacting to a mutation, for instance, or the absence of a protein that is very low, even if we don't detect that protein directly, we should see the effect of this presence, of the absence, or the mutation of this protein on the overall proteome. The argument would be that if we have a mutation, even in a protein that is present at 100 copies per cell, which we would not see by the DIA, very likely, that if this protein is somehow screwed up, mutated, or absent, that we would see, so to say, the echo of the absence of this protein, how the cell reacts to it in the 99 or so % of the total protein mass covered. This has actually not been proven. It's an assumption, but it is the justification we would use that we can generate biologically valuable results on this level of 4, 5, 6,000 proteins covered. Even though we see not all proteins, but we would assume that virtually for every perturbation we would see some form of echo, and otherwise we would have to postulate that there's an entire biology that is occurring at a very, very low level, and we have not seen that. Virtually every protein modification, every perturbation always also shows up in high-level proteins. So that's the justification why we think that these DIA methods are highly irrelevant for measuring the response of cells or tissues or humans to certain perturbations, even though they do not see the whole proteome. Okay, now we assume that we can detect now a certain number of proteins, and now we want to ask, if we had this budget, certain number, let's say $1 million for a large study, how should we spend that? Should we try to analyze a large number, hundreds or thousands of samples, or relatively few samples with very, very deep down? So this is now the work from Ting, and we put this in the context of a biomarker study, where we say we like to have two groups, like healthy individuals and disease individuals, and we would like to find proteins that separate these two groups apart. It's a very common approach. It's very widely used. And the question is more subjects or more proteins. So this is the analysis is based on the real data, but on the structure of real data, but the analysis is basically simulation. So now I'll explain what Ting did here. She took a number of data sets that are published from various types of experiments, DDA experiments, a SRM experiment, and a SWOT experiment, where a number of protein samples were analyzed by the respective techniques. And then she learned the behavior of these proteins in terms of the variability of these proteins throughout the measurements of the cohort. And as you probably know, as those who have studied analytical chemistry, any analytical method is always doing quite well when, in terms of variability, when you have high signal to noise, and it gets difficult when you run into the signal to noise limit, then things become more variable. So this is the behavior. So what Ting did, she plotted basically the standard deviation of these proteins onto the y-axis and the intensity on the x-axis, and then she broke this down in quintiles, basically in 5 areas of proteins which are behaving in that abundance range. And as we can see here in the DIA technique, we see the typical behavior. The high expressed proteins have low variability, and when they get very low down to signal to noise, the variability goes up. This is not true for SRM. And the reason, of course, is not that this breaks this kind of analytical law, but the proteins for SRM are usually highly selected to be well-behaving, high signal to noise. So they're pre-selected by the person who measures them. So they're, in that regard, not a typical analytical technique. Of course, if you go very low down, you will also see noisier data. And this is data from the DDA. DDA analysis, and we see it does the same thing. goes up, but then goes down, and we think this is statistical error because then you're in the basically stochastic domain. This was done by spectral counting, where you counted how many times is a peptide detected over in the data set, and you can either have 0, 1, or 2, and so this is no longer like a accurately quantification. It's basically a stochastic event. Okay, she then did this for other data types, and I want to point you to this lower corner, which is very interesting. This is very relatively new data from a high-end trapping instrument, a Lumos instrument, not to advertise this Lumos instrument, but the noise behavior of the data generated from this type of instrument is absolutely amazing to us. And you see here that the range, the dynamic range, these are log2 levels of protein abundance measured by the signal intensities. You see it spans along a wide range, and it also does not really go up that much like the older instruments, although we also have to say that in this data set here, it includes not just technical variability, but also there's a lot of biological variability because different cell types were analyzed. But in any case, what this shows is that the field is progressing with improving the instrumentation to generate increasingly very, very nice data from the point of view of the noise behavior and the number of proteins covered as well. Okay, now that was just a side note to say that the field actually is progressing quite rapidly also from the data acquisition side. So now to do this simulation, to answer the question I posed, more proteins versus more samples, what Ting did, she broke these data sets with the specific behavior of the noise learned from real data down into a computer, and then she starts to do simulations. So for instance, she would put predictive biomarkers in, so synthetic proteins in a computer, simulated proteins, which would have certain behavior. They might be present in one type, like in tumor, or in case and not in control, and they might be spread out either all in a high abundant level, low abundant level, or spread out over all abundance level. So this is the type of simulations that she can do in a computer. And then she can ask, when she would now add more samples or more proteins by inference going deeper down into the proteome, what is the effect of that on the ability to separate these groups, case and control, apart? So this is basically what she did. She has training, she trains a classifier, and then she evaluates the performance of this classifier in a validation set. This is what one would do also in the real world in a biomarker study. You would like to use more proteins you have maybe identified from the literature or from some source and test this prediction in a validation data set, except she can do this very fast in a computer with hundreds of simulations under different conditions. So now the point that I want, so there's then measures to say how effective is a certain data set that either has a certain number of proteins or a certain number of samples in it under the noise behavior of the actual data that were learned from a data set, and we can ask, how do we gain optimization? So always darker numbers here is improvement. We like to be at value 1, which means we have great ability to predict whether a person would be a disease case or a control case, and it will also give the weight whether the proteins that are detected as biomarkers really have value to be biomarkers, roughly speaking. So what we see here is we have the number of subjects on the y-axis, number of proteins on the x-axis, and we see in this simulation that always, and actually in all simulations, that always the number of subjects helps. It is beneficial. The number of proteins added is not beneficial. In fact, it's detrimental. This is counterintuitive to some extent. But you can understand this very easily if you were to say, I'm coming in from this side here, from the right-hand side where you have high signal-to-noise proteins, and then you keep adding more proteins, and these will be the new ones, will be increasingly low abundance with high variability. So you basically start to dilute out signals that you might have seen before by noise. And so the upshot is, this is skip that, simply more data. The upshot is that if one, and this is an important lesson I think from the point of view of the DAA techniques, if we want to do classification, but I think this also applies to other type of analysis like clustering, although I'm not sure about that, but certainly in the case of this biomarker scenario where you want to separate groups, it always, under any condition, with any instrument, with any data type, and with any kind of with any kind of simulation, it was always more beneficial to make a larger cohort than to go very deep in terms of proteins. So this means that for those who are technology developers, we need to spend our effort to speed things up, that we can do relatively easily hundreds or thousands of samples. We need to do them highly precisely. And for the data scientists, the data analysis scientists, it means we need to be able to analyze this data in a way which does not introduce artifactual or artificial distortions. I'm coming back to that in the last few minutes of this talk. So, more proteins decreased predictive accuracy, more samples increases predictive accuracy, lower technical variability is beneficial, and so as you said, we should focus on, for those of us who work in this field, we should increase the number of samples, even if it comes to some extent at the cost of proteins covered. Of course, we like to do both proteins and samples. Okay, now I said before that I will come back to this issue of does data science alone, large data set as we generate them by, for instance, these DIA methods, we now have established that we have a great resource if we have large data matrices with lots of samples. I would like to ask, are these data by themselves useful to learn something about biological mechanisms? So when we talk about big data sets, which these matrices represent, the most frequent type of analysis is some form of correlation, where we have independent variables, let's say here 2, and we observe them under many conditions, and then we can see how they associate with each other. So this might be trivial things like the more we eat, the more sluggish we get, or I mean, it could be make up any kind of example. And we use this, of course, often, and the whole world of the Facebooks and Google is substantially based on such associations. They have various variables and try to learn something how they go with each other, and then you somehow link them together. So this brought out the notion, which I found once. It's now relatively dated, but this is an article that was published, and which I caught my eye and said the end of theory, the data deluge makes the scientific methods obsolete, especially in the life sciences. So what does this author mean? He said that we were used as biologists or clinicians, we are trained to develop a hypothesis based on prior knowledge, and then to test this hypothesis with an experiment. And if we do this well enough over time, we would learn something about the biological mechanisms that are at play in a cell or under specific conditions. And essentially the whole scientific literature in the life sciences is based on this approach. And he basically says we can throw this away, and we simply have to accumulate data and do correlations, and so that is enough. And eventually the mechanisms that underlie biological systems and processes would also emerge. So basically, what he says, if you live work in a biology institution, department or school, And I mentioned at the very beginning that we have these departments or units which are traditional molecule and function-oriented, mechanism-oriented, like cell biology, biochemistry, genetics. He basically says, forget about all those, close them down, and go just to this side over here. That's the prediction. And now, this is actually a very significant prediction, and I think it deserves some thought. Because if this is true, we of course have to radically rethink how we do research. If it's not true, we should probably wait a minute before we shut down our biochemists and cell biology and structural biology laboratories. So actually Olga and I have discussed this now for a long time, and we then did something about this, and now I want to point out, make a couple of points here. So we wanted to see that if we generate data sets, which is basically a data matrix, whether we could augment this, what we can extract from this data matrix, if we bring in prior information that is represented in biological mechanisms. This is the idea. Do we still need the biological mechanism as prior information, or is the data by itself enough? So we link proteome measurements to genotypic and phenotypic variation in a genomically well-defined system. This I explain now what this does, and then we try to use this data to learn something about the mechanism. Okay, so the system we work with is an amazing system. It is not created by us, of course. This took years to generate, and it's a family of mice. It's called the BXD family, and it's also referred to as genetic reference panels. So here we had at one point, the group that generated these mice had at one point 2 laboratory strains, a male and a female from C57 and DBA. These are 2 common laboratory strains. And many experiments are done in inbred mice of these 2 strains. And then they were crossed, and then out of this initial cross, through crossing mechanisms which I don't want to get into, about 150, in fact about 180 strains were generated. And they are all animals within one strain, are genetically identical. They all have in common that their alleles, their genes, are either from this 57 or the DBA mouse, but the alleles are spread out, basically shuffled around at random through recombination. So we have like a really large family where genes from 2 founders, mother and the father, are spread around, the alleles are spread around at random in 180 offspring, and each one of these offspring is basically present as in multiple copies. So I think this is a fantastic resource, because if we cannot really explain biological phenomenon or clinical phenomenon in those, we will have little chance to really explain them in an outbred human population, for instance. Because you can eliminate all confounding factors like age or food intake or whatever, because you can keep, of course, these animals under exactly the same conditions of the same strain or across different strains. Okay, so these animals have been extensively phenotyped, again by this BXD consortium. And so, for instance, measured how much their physical prowess, how much they run at night, how fat they get if they eat fat food, how long they live, hundreds of phenotypes have been measured. So what we wanted to see is if we could use this resource, basically do molecular measurements in a somewhat big data situation, and relate this to phenotype and learn something how the 2 are connected. So the study design was to take these mice and then which is a genetic axis, and then to put these mice, this is the genotype axis, and then to put these mice into different environmental conditions, or feed them with different food, some that get fat and get diabetes and eventually die, and others that do not. And then we identified, measured a number of proteins, in this case by SRM, but the same thing we've done also by DIA, but it doesn't really matter. The point I want to make is the same. So we could not measure all proteins. We could also not measure all strains, simply for throughput reasons. Now we probably get into a stage where we could do more of all the strains. So we measured 40 strains under two conditions in duplicate, and what was measured was a number of proteins which are centered around metabolism. So it's not a random selection of proteins. The proteins which have already some functional significance. This shows, so these were 192 proteins were targeted across these strains, and so this shows the technical CV, technical replicate CV, biological replicate CV. Technical means the same sample is injected twice or 3 times, and biological replicates means the same mouse or the same liver or the same brain from this mouse was overall the whole process was processed. So we see that the technical and biological CV within the same strain is very tight. These are high quality data, and when we then look at the whole data set and ask the CV of peptide ratios across the mice, we see a much larger or substantially larger CV. So we will conclude that the difference we see between the repeat analysis and what we see in the overall data set is actually biological signal, not just experimental noise. So that's what we assume from that. So this is the data matrix, not very large data. It's 192 proteins measured over close to 80 mice, and so this is the data set. So now what we can do with this data set, we can ask, yes?
In fact, you're using a ratio, does that mean you have injected reference standards for all peptides, or you have isotope label standards?
So this is just based on the integrated peak area of the peptides.
What's the ratio?
The ratio.
Yeah, what does it mean, the ratio? Is it label free?
It's label free.
Okay.
Yeah. So it's basically the intensity, and then it's probably normalized to the mean or something. Okay, so what we can do with this data, and we can ask, this is now an association question. We can ask if we know, of course, which allele is present in each mouse at a particular locus, and we can now ask, is there an association between the amount of a protein or a peptide with a specific allele? So when you have in the population 2 alleles, a BLAC6 and a DBA allele, and you can ask, is it so that whenever the DBA allele is present, a protein is high, and whenever the BLAC6 allele is present, the protein level is low? Then and if this is happening over all these mice, then you have a strong case to say that the genetic locus, the allele at the genetic locus, is linked to the abundance of the protein. We don't know, of course, how. There's no mechanism behind it, but it's a statistically supported link. Okay, so this is referred to as quantitative trait locus mapping, and we measure this. We call this here protein QTL, protein quantitative trait locus, and this basically means that we identify an association between an allele at the specific locus and the abundance of a protein. Many have no such association, but some do, and we see that we have a number of proteins which are which have such a protein quantitative trait locus. Some act on themselves. For instance, you could imagine a transcription factor acting on itself, or others act on something else in a mechanism we don't understand. But there is a statistical link, and this is what, as far as we would get with this kind of no hypothesis driven approach. We basically can take various variables, we can correlate them, and we can successfully come to this. This is kind of an endpoint. We don't know how we would go in a data-driven way further than these associations. Now we would like to see whether by invoking prior information, whether we can learn something about the mechanism. So this is now an example, actually quite a nice one. There's several of these, but this is the one I think is very plausible to understand. So one of these QTLs is for an enzyme that is at the last step of the degradation pathway of amino acids, specific branched chain amino acids. Doesn't really matter in detail, but you know that, of course, every cell has a metabolism. It builds up molecules and breaks down proteins, and these pathways usually are sequential enzymatic steps. This happens to be the case here with amino acids like lysine. They get sequentially degraded to eventually acetyl-CoA. And one of the enzymes that is down here, which converts this step down here, is one for which we found a QTL. The enzyme is called DHTKD1, doesn't really matter. But so what does that mean, what I'm saying now? It means that there is an enzyme in this pathway, and this enzyme can be either, for genetic reasons, be in a high amount in some mice, based on the allele that's present in the genome, or it can be in a low amount in other mice if the other allele is present. And we assume that the amount of this enzyme present will be a determinant how much flux there is through this pathway. You can think of it like a water hose. When the water hose is large and wide open, a lot of the enzyme would be present. The flux is high, and if the enzyme is at a low level, it's constrained, the flux is low. So if the flux is low, we should see a pile-up of the upper of these metabolites here that are the product of intermediate steps of this degradation pathway. So we measured those, and these are two of them, two amino adipate, and it doesn't really matter what they're called for this discussion, but it's two sequential metabolic intermediates which you can measure, and we could see then we correlate these, the amount of these the amount of these two metabolites with each other, or with the amount of the enzyme present, which is controlled by the genetic locus that we identified by association. So what we see here is the water hose effect. If there is a lot of the protein present, there's a lot of flux, and we see little amounts of this intermediate metabolite. So it kind of piles up. If we correlate the two metabolites by themselves to each other, they go synchronously. So this clearly means that the genetic locus that causes this protein QTL has affects the flux through this metabolic pathway, because if it's low, the metabolites pile up. So now then we could find by chance in the literature that this amino adipate is a biomarker for diabetes risk. So now we make a link which would never have been discovered. This is out of the Framingham heart study, a long-standing study that's been coming out of Framingham. And what we would never have predicted this, probably made this link without having prior information. So simply by looking at this data that came out from the measurements, I think we would never have gotten this. But we got this, that we could extend the initial statistical association of QTLs to metabolites. We could associate them then with show that they regulate the locus effects through the mechanistically through the expression of this enzyme modulates the abundance of certain metabolites, and these metabolites have a role in a human disease. So I think this is an example that we would say that correlation is not enough. Correlation is great to point us to certain areas, and then I think we need to get into other biological methods, including those which are present in our departments, biochemistry, structural biology, to explain what really is going on. And I think the strongest departments or units will be those which can really combine the enormous power of both of these approaches in a sensible way around the biological question. So the conclusion is, we came to the conclusion through that and other studies that it is not enough to just correlate if you really want to get somewhat deep insight into biological mechanisms. So now I have the last 2-3 minutes. I have a couple of cautions. So we need to pay. We now established. I think that we want to generate increasingly large data matrices with lots of proteins, lots of samples, and that these really have useful information that we can mine. Now we have to be cognizant that there is a lot of problems remaining, and if you don't pay attention, we fall into traps. The one I want to point out is false positives, the other is batch effects. In neither of them I'm going into great details of this very fast. So I mentioned at the beginning the two studies from the with these proteome draft papers, and I said that they overestimated from the data the number of proteins that were probably truly in the sample. So we analyzed this effect, and it is an effect that I think is now fairly well understood. And Manfred Claassen, who was at the time a PhD student in our group, analyzes and wrote eventually a program that should correct for this. So what are we seeing here and what is going on? Let's just say you have one sample, you make an extract of a cell, and you do a shotgun experiment. You identify a number of, you generate a number of spectra, and you associate this spectra with a peptide sequence. It's referred to as peptide spectral matching or the sequence database searching. So you will do this with your sample, and you will dial into the search engine something like you allow 1% false positive identification. So 99% of these assignments would be correct, 1% is wrong. So when you do this once or with two samples, you will see that you start to, this represents the proteomes, would be like 20,000 orbs, and you will identify a number of proteins which you, let's say you have identified with with a false discovery rate of 1%. So this is quite accurate, and I think we are all very confident that we can do this well. And I'll come then to the DIA issue just in a second. This is DDA situation. But now let's say we do it another time, and we do it 100 times. What we observe then is that the proteins which we know already are in the sample will be identified with an increasing number of spectra because we know they're in there, they're true, they're always within these 99% true ones, but if the false ones, the 1%, there's no reason why they would associate with any specific protein. So by they're simply randomly distributed over all the proteins of the genome. So now when we do this long enough, we will see, let's say we do this 100 times, analyze the same sample 100 times, we will see that we have hundreds or thousands of repeat analysis, identification of the same peptide, but we have eventually then a false positive, a false hit to any conceivable protein in the proteome. Simply because this 1% or 0.1% of false positives, they will randomly distribute. There's no reason why they accumulate on the true positives. So they will eventually, if you do this long enough, you have hit, seemingly identified every conceivable protein. So the upshot is that if one does this analysis by increasing the number of repeat analysis or peptide spectrum matches, if he increases those here, basically adds data to the large data sets, you will eventually see a curve of protein identifications that starts creeping up. Eventually it hits 100% of the proteome, and what it really does, it will cap out, and the true positive will cap out and basically will not increase. So the problem of large data sets, or one problem we have to always watch for, is that if we keep adding data to a data set, so when we extend the matrix on the sample side, we should always see check whether the curve of proteins identified, whether it's in the DDA world or DIA world, doesn't matter, whether it keeps creeping up or whether it stabilizes up. If it keeps creeping up, there is almost certainly some problem with the controlling the false discovery rate. So this was also observed in DIA, and several groups, in fact, several groups got together and analyzed this and provided a solution, including actually Mike's laboratory. Brendan is on this paper, groups from Mike Koss and other groups who are really worrying about these things. And I don't want to really go through this figure in great detail, but it is exactly the figure that what I tried to show before. Now in reality, it shows the number of runs that would be done in DIA world. I'm actually coming back to this data on Friday. These were about 230 repeat runs of exactly the same sample. And we initially observed that if we did not specifically address this issue, that the number of proteins identified or or peptides, doesn't really matter what it is, peptides or proteins, just it will go creeping up. And this was a sure tell sign that something is wrong with the control of the false positives over the whole data set, not within each individual run. And then the number of people named here, specifically George Rosenberg, generated a system, a controlled system for controlling the false discovery rate. And the upshot is that if you apply this over even very large data sets, we see now the curves nicely being stable, so we can add another 100 runs, and the number of proteins that are wrongly identified remain stable and will not be creeping up. So this is a sign, this is one of the traps that we tend to fall in with large data, that we can no longer or do not consider the implications of controlling the false discovery rate as an aggregate over the whole experiment. And if maybe, I don't know whether this is going to be discussed in the course of it. The other trap is batch effects, and here I show an example. This is someone in our group unearthed that it's an interesting example. So this is the mouse ENCODE consortium. So they measure gene expression data by gene expression analysis, and they compared. The question is an interesting question. When you compare gene expression patterns in a number of human tissues, in a number of mouse tissues, whether the tissue, the same tissue, say liver, mouse liver, and human liver would be closer together than to each other across species than to tissues in the same animal. And they came to the conclusion that this is so, that it's the opposite, that all the tissues in the same species cluster together more closely than across species. So the mouse lung, liver, brain clustered to each other more closely than to the analogous tissues in human. So then this was published, actually in Nature, and then another group reanalyzed this data, and they came to the opposite conclusion. So they came to the conclusion that this was not so, that in fact mouse liver and human liver was more close to each other than, let's say, mouse liver to human brain. I mean, it's fundamentally a reversal of the conclusion. And they concluded that they analyzed this, and they said that this group, the initial group, introduced the systematic error, but in the way they aligned samples in the flow cell for RNA sequencing to generate transcript maps. So they introduced an experimental confounding effect, a batch effect, because they grouped some batches together, and then basically what they measured is the effect of the flow cell on the results rather than the biology. And they said here, this is a statement they make, these other authors, they said their conclusions are unwarranted, they're not wrong, because the study design was simply not suitable to addressing the question of tissue versus species clustering of the gene expression data. So what I'm trying to get at here is that we have to be very careful with these large data experiments, that we try to recognize if we have confounding effects or batch effects, and if we try, of course, try to avoid them, but if they exist, we should then try to correct them. So here the experimental design was such that there was not recognized there was a technical effect, basically, that was then interpreted as a biological effect, and so that led to the wrong conclusions. So now, what are possible batch effects? There's many, too many that we can even think of. This is just a short list. Could be that the mass spectrometer gets cleaned between runs. Could be that a student goes on vacation, someone else continues with the experiment. It could be that there is a lot of a reagent runs out in the lab and we order a new lot. I mean, these batch effects are inevitable. They are simply not avoidable. We can minimize them. We can try to minimize them, but we are not able to completely eliminate all variables. So the only chance we have is to design the experiment such that such batch effects would be recognized and could be then eventually corrected. If you don't recognize them, like in this mouse versus human case, we cannot correct them. because we don't know what we should correct. So then there is, this has huge implications for experimental design, and I think Olga, I just asked Olga before this will come up in the course of this module, and I don't want to spend a lot of time on that, but one of the important things here is to structure the samples throughout the analysis such that that effects on batches can be recognized. This is an example of a study we did in our lab where we had two cell lines that were subjected to various drugs and over various times, and so this created 200 samples, which is rather large for proteomics at this time. And so the way this was done is to put these samples into various batches where when the experiment was done, the person who did the experiment, the mass spectrometer who ran it, were separated into various batches so that we could would detect if there's a systematic problem with one of these variables throughout the experiment. So this is referred to as well. So with that, I would like to finish and basically say that I hope that what I try to convey is that we are entering a very interesting world in proteomics where we can really generate these large data sets. We think that we should focus on getting large numbers of samples through because that's where the information lies. We should try to find ways to integrate these data sets with prior biological knowledge. This is, I think, the trick to really learn new biology. And even though if we get excited about the ability to do large-scale measurements, we still need to be somewhat cautious and cognizant of some traps we can fall in. Two I mentioned, batch effects and the accumulation of false positives, which seemingly make the data much better than they actually are, but it's better to have sparser data than large data with a lot of mistakes in them. So thanks for your attention, and I'm happy to answer questions. Someone have a question? Okay, great. Then it's probably. Oh, you have one. Yeah.
So there's a comment. I think your DIA methods definitely have a lot of promise, but you yourself brought the point that a lot of false discovery rates are there. So is there still a way out that we can say which method can be much more robust eventually?
Whether the technique can be made more robust? Yes, I think it's getting more robust all the time. As people learn by doing, and I think we are one of these people, and we constantly learn. I think the problem areas are shifting. I would say now that when DIA started, the issue has a lot to do with mass spectrometers. I think that's what's concerned us, the chromatography. I think this is not perfect, but I think this is fairly well understood. And now I think we see most of the variables come from the sample workup, the sample processing, and I think there's certainly an area where a lot of kind of standardization can be generated. And so we use, for instance, this pressure cycling technology, but there's other ways to do that. But I think it's very important that the digestion and protein extraction is done in a consistent way, and that the best is, of course, if a machine does that, because the machines are more consistent than an individual persons. And then I think we also learned a lot already about the data analysis. I mean, we were initially failed also in this trap of this FDR accumulation, exactly like in EA, and we fortunately recognized it, and then this was corrected. I think it's sensibly corrected. I wouldn't say it's perfectly corrected, but Ben will talk more about that. And I'm sure there's other issues, but the instruments also get a lot better. The resolution gets better, which helps the accuracy of the results. And so.
So major variability is at the sample prep level now more as compared to the technology.
I think this is the area probably where presently the most gain could be made when you have 200-300 samples, let's say tissue samples, to process them in a way that they really are consistently processed. I think is still a challenge.